Hello students, today we are going to discuss the last part of the chapter biomolecule, that is the enzyme. So in this part what we have to know, see, one more thing is very much important, that this part enzyme, from here one question will come. Okay, so in NEET you have to prepare such a way that yes, this is important and some conceptual parts are here, and the questions come from the KM, VMAX, and people used to confuse in this. And in the NCERT some words, some sentences are given, and those sentences if you cannot understand, you will not able to answer the questions. So that's why we will see each and every lines of NCERT. I have pointed out all those parts, and we will discuss everything in this video. So let's just start. So first of all, in this enzyme, some characteristic features I have to tell you. First of all, to know the enzymes before coming to their actions. So first thing about the enzyme, the basic features or characters, if I need to say, that these are protein in nature. So very much important thing, that earlier in this course of evolution, earlier the RNAs were used to be called, or it was evolved such a way to act as the enzymes. So later on, whenever evolution took place, then nature selected the proteins to act like the enzymes. So what are the enzymes? In our body, number of biochemical reactions go on, and those biochemical reactions to be carried on, we require some helper or some catalyst, and those catalysts in biology we will term it as the enzymes. So chemically the enzymes are they are protein, okay, and generally they are the tertiary protein. Okay, so I have told you in the earlier video, that the proteins may have different structures: primary, secondary, tertiary. So what happened, the enzymes, whenever it forms, earlier it was linear, but gradually folding occurs, and these foldings are going in such a way that at one period of time, whenever it attained the tertiary structure, it becomes functional and functional to carry out different biochemical reactions. So these are protein in nature, which possess the tertiary structure, and generally these tertiary structured proteins are the enzymes who catalyze different biochemical reactions in our body. Second very much important thing, that I have told you, that structure wise the enzymes, these enzymes are of the size globular structure. Okay, the proteins can be of two structure. One is globular protein, another is fibrous protein. So fibrous proteins are insoluble in water, but globular proteins, the enzymes are globular proteins, they are soluble in water. So one very much important thing you need to know, that they are soluble in water. Okay, and always remember, whenever enzyme works on the substrate, it always works in the aqueous medium. Aqueous medium means where water means watery solution. So in the water only, how the enzymes can work? By the process of diffusion. So diffusion occurs, enzyme attaches to the substrate, and then it forms the product. The next very much important thing, that it functions in specific pH and temperature. See, any type of enzymes to work, it require a specific pH. In the digestive system, we need to know that different enzymes are present to carry out the process of digestion. Suppose in our mouth, we have such enzyme. So in the mouth, in the buccal cavity, we have a specific pH. Okay, we have to maintain a specific pH. So that 6.8 pH is the pH where the enzyme salivary amylase works. In the stomach, we have a specific pH, that is 1.8 or 2 pH. In that pH, only pepsin enzyme can work. Okay, so every kind of enzymes require a specific pH where they can work. I have told you that it maintains the tertiary structure. So this tertiary structure is a balanced structure. It can be maintained balanced only at a specific pH. Whenever the pH changes, then the tertiary structure also tend to change. So the enzymes cannot work. Okay, next one, it also works at a specific temperature. Okay, our in case of homeotherms, if I need to say, the homeotherms, our body temperature is 36-37 degrees Celsius. So in the 37 degrees Celsius, our most of the enzymes work. Okay, so there is developing, the enzymes of our body is adapting itself to work at a specific temperature. If the temperature changes, right, so the enzymes may be inactivated or maybe it can be of denatured, right. Next, very important thing, that if it comes to the enzymes, okay, enzymes generally pauses. Okay, the another point I need to tell you, the enzymes, okay, the enzymes work, or enzyme attach to substrate with the active site. Okay, so just see what happens. The enzyme, suppose this is the tertiary structure of a protein. Okay, tertiary structure of an enzyme. Now what happens, whenever this tertiary structure develops, the enzyme is developed, then on the surface of the enzyme some pockets are developed. Okay, suppose this is a pocket, this is one pocket, this is the next pocket. So this way such pockets are developed. Okay, or crevices are developed, that is given in the NCERT. Some crevices means some groups, some deeper regions are developed. So those pockets like structures, they are called as active sites. Okay, in this active sites we will have some variable group. Suppose here it is a variable group, functional group is present, and with that functional group we can add a specific enzyme. Okay, so we can add a specific substrate. So here suppose the substrate is there, so this substrate can be added chemically with the variable group, and it will be added or it will be fit into the pockets. So that pocket or that crevice where the enzyme can be attached with the substrate will be called as the active site. Okay, so this will be called as the active site. Active site is the crevices or the pockets developed on the surface of the enzyme with which the substrate can be added or substrate can be attached, so that the chemical reaction can be carried on and the substrate can be converted into product. Okay, that will be called as the active site of an enzyme. And we need to know that the enzymes are going to be very much specific, because with that crevices or that pockets we will have some specific functional group or specific R group. With that R group, the substrate will also have the specific R group which is going to be reacting with this enzyme. So that's why these enzymes are said to be very much specific. Any of the substrate cannot be attached with the enzyme. Enzyme have to be very much specific, and that enzyme will work on only that specific substrate, right, to carry out the reaction to produce the products. Okay, next, as I was telling you that, yeah, third point, that the enzymes functions at specific pH and specific temperature. Yes, so our body is adapting ourselves to be called as a homeotherm. We have a specific temperature, that is 36.5 or 37 degrees Celsius. So in that 37 degrees Celsius, what will happen? Our enzymes work. Okay, so that's why these enzymes needed to be maintained, okay, in a specific temperature, and only in that temperature enzyme works. Yes, some exceptions are there. So what are those exceptions? One example we have, that enzyme can work in a very high temperature also. Okay, suppose one example we have, that is Taq polymerase. So that Taq polymerase you will get in the second year in a biotechnical procedure we have, that is PCR, polymerase chain reaction. In the polymerase chain reaction, we require one enzyme. That enzyme is Taq polymerase or DNA polymerase, which can be active up to the 90-92 degrees Celsius. In that temperature also it can work. So this obviously enzyme is not ours. Okay, this is not extracted from your body and used in the PCR technique. So this Taq polymerase is actually acquired from some bacteria that is called as Thermophilus bacteria. Okay, so it works in a specific pH and temperature. Okay, I was telling you that the enzymes are protein in nature. So what can happen in the nature? I told already that it was the RNA enzymes also. Some of the RNA enzymes are there which are not protein in nature. Normally most of the enzymes are protein in nature, but sometimes it can be different also. Some exceptions as I am telling you. So here also they are protein in nature normally, but there are some exception. Okay, what is that exception? Some RNA enzyme. Okay, and those RNA enzymes, one of example is ribozyme. Okay, so this ribozyme is not made up of protein. It is not chemically protein. Okay, it is ribozyme which is not chemically protein. Okay, so these points I hope you understand. First thing I have told you that the enzymes are protein in nature. Second thing, they are generally globular protein. That means it is soluble in water. So enzymes work in aqueous medium. That point you need to know. This is also very much important. Third point, that it functions in specific pH and temperature. Above the pH or lower the pH, in high temperature, in lower temperature, the enzymes cannot work. One example I have given, that except human body or except the homeotherms, what can happen? Some bacteria possess, every organisms will have their specific enzyme. One bacteria is Thermophilus bacteria. The Thermophilus bacteria can survive in very high temperature, so their enzymes also work in a very high temperature. It is around 90 to 92 degrees Celsius also they can work. So that enzyme Taq polymerase is isolated to be used in different biochemical procedures, that is PCR, polymerase chain reaction. Some techniques are available, we will discuss. Next one, the next important point, the fourth point I need to tell you, that rate or a speed of a reaction can be increased by the enzymes. Okay, so I have already told you that in our body some biochemical reactions goes on, and those biochemical reactions, whenever it is carried on, then enzymes, if it is added, then the reaction gets faster. Okay, so the rate of a reaction can be measured, it can be achieved, we can achieve by adding the enzymes, and whenever we add the enzyme, the reactions get more, or reaction gets faster. Okay, so the rate of a reaction can be measured by the amount of product by the time. Okay, so what is happening? See, what is it, that the rate of a reaction, okay, it is actually one reaction we have, the reaction, just see. So here what is it, suppose one reaction we have, and that reaction occurs in the human body, in the RBC. So in this RBC we have one reaction, that is water that reacts with carbon dioxide. Okay, it forms a compound that is called as carbonic acid, and this reaction occurs in absence of enzyme also, in presence of enzyme also. So if there is no enzyme, okay, then in 1 hour only 200 products are formed. Okay, 200 products are formed in 1 hour. Okay, but if you add the enzyme, that enzyme is carbonic anhydrase, and remember, carbonic anhydrase is the fastest working enzyme, okay, of our body. So whenever carbonic anhydrase is present, in presence of carbonic anhydrase, the carbonic acid is formed. This 6 lakh of molecules, 6 lakhs of carbonic acid will be formed in 1 second. Just see how this rate of reaction gets faster. So obviously with enzyme or without enzyme, how the reaction is changing its rate. Okay, so obviously the rate of a reaction here, it will be very much less. Okay, you have to divide 200 by 1 hour, isn't that 1 hour? Converted into seconds, you will get the rate. But if it is the enzyme carbonic anhydrase is added, then the rate of the reactions get very much faster. Just see, 6 lakhs of carbonic anhydrase is produced in 1 second. So here the rate of the reaction gets higher. Okay, so just remember, whenever it comes to the rate of a reaction, always remember that in absence of enzyme, the chemical reactions occurs. In our body, chemical reaction occurs, but the rate of a reaction get decreased. In presence of enzyme, the rate of a reaction get faster. Okay, so one example I can give you, that is the fastest working enzyme, that is carbonic anhydrase. That carbonic anhydrase, faster the reaction that occurs inside the RBCs, that is the water, okay, that binds with carbon dioxide in presence of carbonic anhydrase to produce 6 lakhs of carbonic acid in 1 second. So that is rate of a reaction. So I have told you the fourth point. Next on active side, already I have told you that in the tertiary structure, whenever the proteins get folded and form tertiary structure, to become that enzyme, some crevices or some pockets are developed, and in that pocket the substrates get attached. So what are the substrate here? Water and carbon dioxide, these are the substrate, get reacted in presence of the enzyme and it forms the product. What is that product? That is the carbonic acid. Okay, so I hope you have understood this introduction points. After that, the next point, we need to know that how the enzymes work. We will see the mechanism of working of one enzyme, how the enzymes work. So first of all, I have drawn here, suppose this is one enzyme. Okay, suppose this is one enzyme. Okay, so I have told you that here this one will be the active site of this enzyme. Suppose this is the active, make it a little bigger. So this is the enzyme. So with this enzyme, a substrate will be attached. So this substrate will have the same structure, okay, same shape. Okay, suppose this is the substrate, this is the enzyme. Okay, then after that, what will happen with this enzyme? The substrate get attached and we will have one intermediate or transient state. And what is that intermediate or transient state called? That will be ES complex. Okay, ES complex means this enzyme, whenever attached to the substrate, first of all it will attach to the substrate loosely. So first of all, loose attachment occurs. Okay, so it will have loose attachments. Okay, just be attached. I have told you here the functional groups are there. Then after that, what will happen? Without getting consumed, remember, enzyme will never take part in the chemical reaction. Okay, without getting consumed, this enzyme will change the chemical structure. Okay, chemical structure of the substrate, and the substrate will be converted into product. Okay, then after that, the next step will be, it will be the product formation is going on. So this will be called as EP. So first of all, it is like this, just a transient state has been formed. Okay, so just change it little so that you can see properly. So what is it? Suppose here this is the enzyme. Okay, this is enzyme, this is the substrate, this is substrate. So whenever with this enzyme, the substrate will be attached. Okay, then we will get a transient, this means intermediate state. What is this intermediate state? This will be enzyme substrate complex. Means first of all the shape has to be same. Okay, so this is the same shape, so that's why this substrate is added to the enzyme. So first of all, due to the shape, the enzyme can attach with the substrate. Okay, then an intermediate state has been formed, that is enzyme substrate complex. Then after that, the next step what will happen, that without being used or consumed, this enzyme will change the chemical structure of the substrate. So here the substrate is there, but what happened now, this chemical changes will occur. So here some chemical changes occur. So some chemical changes occurs in the substrate, but there will be no changes occurs in the product. In the enzyme. So this state will be called as enzyme product state. Okay. Then after that, what will happen in the next step? That in this next step, what happens? The product will be formed and enzyme will be released, and now it will be available for the another reactions to be carried on. So each and every term what I have just said is important. So here the product is formed. Suppose this is the product. So these are the products. This is the enzyme. One second. So just see what I have said, that here enzyme attached to the substrate, it form an intermediate state. So this is the intermediate state, means first of all in this state when enzyme substrate complex has been formed, then this substrate attaches to the enzyme loosely. Okay, loosely. Then after that, what will happen next? What will happen? This enzyme attach to the substrate tightly. Okay, tightly, and change chemical structure. Okay, then what will happen? The enzymes will convert the substrate into product. Okay, product formation is occurring, but this enzymes, nothing is going to change in the enzyme. Enzyme will be not consumed. Okay, remember what is happening to the enzymes. Enzyme take part in chemical reactions, actually biochemical reactions here, chemical reactions to form product from substrate without being consumed. Okay, so this is going to be very much important. Here what is happening, that the enzyme will take part in the chemical reaction, but it is not going to change its shape. Okay, so first of all, with this enzyme, the substrate has the same shape, then only it is possible with the enzyme substrate can attach. Okay, so there are a number of compounds they are present. Okay, might be possible that different compound can also have the same structure. Okay, same shape with the substrate. Substrate is looking like this. Suppose there is one also compound, but it is going to have different shape. Okay, so it can be attached to the enzyme, but it cannot take part in the chemical reaction. Okay, so what is happening? They will form some intermediate stage, but this stage, whenever it is there, they will not go to this stage. Next EP stage it cannot reach. Only the substrate can be added. So maybe the other compounds will have same shape. Okay, but their chemical structure, obviously the chemical structure is going to be different. Okay, so obviously if the chemical structure is also same, then obviously it can be the same product. Substrate itself isn't that. Another product may have the same shape, but the chemical shape or chemical structure will be different. Then what can happen with this enzyme? The another compound other than substrate can be attached, but they will not change its shape, or it will not take part in this reaction, or the chemical nature will not gonna change, right? Then what can happen? The reactions cannot be proceeded. Only the substrate can be added. First of all, due to the shape. Second, due to the variable, the group R group, they will take part in the chemical reaction, and without being consumed, the enzyme will able to change the chemical bonds in that substrate, convert into the product, and after that product will get separated from the enzyme. Okay, so this is what happens. Just need to be remembered. This is very much important. So everybody knows this point. But the very important thing you need to know, that whenever this happens, whenever I have written this, I have added this terms. You have seen this is the reversible sign, this is irreversible sign. I have added here. That means whenever it comes to the enzyme substrate complex, yes, one substrate can be added to the enzyme. What I have told, that it loosely attached. Okay, this is the loose attachment. So that's why this one, after that, whenever ES complex has been formed, then maybe it's possible substrate can be once again removed. Maybe possible. Okay, then yes, this state will be irreversible. Oh sorry, this is reversible. Okay, but whenever enzyme product is been formed, that means now they are chemically involved. They are forming the tight attachment. Okay, so whenever just they tightly bound to each other, enzyme bound to the product, means the substrate tightly and converted into the product, then this state, EP state, will be irreversible. Okay, this is irreversible. After that, you cannot remove this thing and can get the same structure as substrate, because already the chemical bonding has been changing in the substrate. That's how it is forming the product. And once this product has been formed, obviously it will be irreversible. Okay, so this came in need, that what is about the ES. ES is intermediate state. So both these two are intermediate. Okay, both these two are intermediate state before forming the product. But whenever it comes to the ES complex, E dot S, then it is reversible. That means once ES is formed, ES complex is formed, maybe it's possible that once again the substrate can be removed. But once EP is formed, after that the reaction is already proceeded, already the substrate has changed in chemical bonding, lot of chemical reactions are going on, then after that you cannot reverse or cannot get the substrate back. Okay, so EP complex is irreversible. After that you cannot get the substrate back. And obviously whenever the product has been formed, already it is detached from the enzyme, then obviously after this product has been formed, you cannot get the substrate back. Okay, so this is how the reaction occurs, and this state, this will be called as the intermediate state. So here one term we need to know. We require a minimum energy to carry out the reaction. Okay, so I have told you that the enzymes, they increase the rate of a reaction. Okay, so here we have a term that is called as activation energy. Now what is the activation energy? Activation energy means it is the energy required by the substrate to convert into product. Okay, so every substrate require a minimum energy. It will go proceed, then it will reach the enzyme substrate complex, means energy level barrier is already been reached. Then after that EP is been formed, then also another level of energy will be required, then after that product will be formed. So certain amount of energy will be required by the substrate so that it can be attached to the enzyme. Then after that it requires the next level of energy so that that the chemical bonding, chemical structure can be changed, then after that product will be formed. So that minimum energy required by a substrate to convert it into the product will be called as activation energy. Okay, so this will be called as activation energy. Remember this thing. Now, we need to know, we have to remember it with the help of a graph, and that graph is going to be very much important. What is this graph here now? Okay, suppose here we have a substrate. Okay, so here I will write here the potential energy. Okay, so first of all, the substrate require a level, a potential energy. Then after that, what will happen? It will cross the barrier of this energy level. Then after that, it will form the product. Okay, so a certain level of energy will be required by one substrate to convert into the product. Okay, so you have seen here, this will be the energy and this will be called as the activation energy. Okay, so this activation energy, okay, is without now the enzyme. So this reaction is going on but without the help of the enzyme. Okay, then also an activation energy will got by the substrate to convert into the product. I have told you the example of water and carbon dioxide. So without the help of the carbonic anhydrase also product, carbonic acid A H2CO3, this product will be formed. It's not like that without the help of enzyme any reaction will not happen. It's not like that. Reaction will go on, but what will happen, the product formation will require more time. That means to gain the activation energy it will get more time. Okay, but whenever you add the enzyme, okay, then what will happen, activation energy will be lowered. Okay, because it will give that certain energy, and that's why what will happen, easily the product formation will occur. Okay, rate of reactions get increased and it will decrease the activation energy. Okay, so here activation energy will now get decreased. So it was without enzyme, okay, activation energy is very high. This is the activation energy, okay, without the help of the enzyme. But with the help of the enzyme, what will happen, activation energy will be decreased. Okay, so just remember, with the help of the enzyme it decrease the activation energy and increase the rate of a reaction. So this point you have to remember, enzymes increase the rate of a reaction by decreasing the activation energy. Okay, so this point is going to be very much important. After that we need to know two very important point. We know already, you have seen already in chemistry, what is the exothermic and what is the endothermic reaction. Okay, what is exothermic and what is endothermic reaction? Okay, so first of all just see what is happening here. Okay, we can give two graphs here so that we can understand how this exothermic and endothermic reactions can go on. So let's just see. So after that, let us see the energy level differences between the substrate and the product. Okay, so if the substrate and the product that are being formed, okay, so you have seen the substrate is converted into the product. Okay, and here in this reaction also substrate is converted into the product. Okay, if the product have less energy level, okay, than that of the substrate, then this reaction will be called as exothermic reaction. Okay, if the substrate has more energy level than the product, then it will be called as exothermic reaction. Why this happens? Because in this reaction heat is generated, so that's why the product have less energy. So as heat is generated, so that's why energy level is decreased in case of the product as compared to the substrate. Then this reaction will be exothermic, and here you can see energy level I have shown here. So this is the potential energy here. Substrate has more energy and product has less energy. So in this reaction the heat is released and the substrate has more energy level and product have less energy level. So this reaction will be exothermic reaction. Second thing, what happens to the endothermic reaction? Then if the substrate has less energy level, okay, less energy level than the product, then what we can call, this will be called as endothermic reaction. Why this happens? Product have more energy because heat is not been released. Okay, so here heat is not released. Okay, heat is taken, it is consumed. Okay, heat is taken, it is consumed, it is not releasing. Okay, so that's why product have more energy as compared to the substrate. So these are the differences between the exothermic and endothermic reaction. The substrate has more energy than that of the product, that's why heat is released, that's why the product have less energy. That will be the graph of exothermic reaction. If substrate has more energy because it is been consumed, it is taking the energy and product is been forming, product have more energy as compared to the substrate, then it will be called as endothermic reaction. Okay, so these two graphs are going to be important for the NEET exam also. After that, let us move to the actions, how the enzymes work, in what specific activities, or how does enzyme work in pH, in specific temperature, how actually it works. Let us move to the next part. See at what are the different factors which are going to affect the enzyme activity, how enzyme work, in which temperature it works, in which pH it works, or what are the things or factors that are affecting the enzyme activity. We will discuss one by one. The first one we will see is temperature. Already I have told you in the introduction that the enzymes work in a specific temperature. We homeotherms are adapted that our body temperature is maintained temperature of 36.5 or 37 degrees Celsius, where most of the enzymes work. So what happens in case of these enzymes? To carry out this chemical reaction, a specific temperature is needed. Below that temperature or above that temperature, the enzymes do not work. Okay, so if you see this graph, okay, so first of all here we will see the enzyme activity. Okay, how it will be working. See, so here when the enzymes are active, suppose this is the enzyme which is showing its maximum activity. Okay, so what is this temperature? So this temperature is suppose 37 degrees Celsius. So in this 37 degrees Celsius, what happens? Enzyme show its maximum activity. Okay, so what is this? This will be the optimum temperature, we can say. So what is the optimum temperature? Optimum temperature means that temperature in which a enzyme shows its maximum activity is called as the optimum temperature. So 37 degrees Celsius is optimum temperature for most of the enzymes in human body. So if this temperature is less, say suppose it is 27 degrees Celsius, and here we will take suppose it is 47 degrees Celsius. So what will happen? That in 27 degrees Celsius the temperature will get decreased. Okay, or in that temperature actually what will happen? The enzyme is inactive. Okay, so enzyme, to carry out the reaction a specific temperature is needed, special energy is needed. Okay, in that energy only enzymes work. So in this less temperature it is getting inactivated. And what will happen in high temperature, suppose it is 47 degrees Celsius, then what will happen? The enzymes get denatured. I have told you that Whenever the enzymes form the tertiary structure, they develop the pockets that are called as the active sites. So active sites gets started to denature, and whenever it denatures, then the enzymes cannot attach to the substrate and carry out reaction. So this will be the temperature at what temperature it is working, 37 degrees Celsius. So it will be called as uh, optimum temperature. Below this temperature, enzymes get inactivated, and above this temperature, the enzymes get denatures. Okay, so just see here we have one term, that is Q10 is equal to 2. Okay, what is it? In NCERT it is given that suppose it is 37 degrees Celsius. Okay, so whenever it is reaching to 47 degrees Celsius, the rate of the reaction gets half. Okay, and whenever from this temperature 47 degree to 37 degree it is moving, then the enzyme starts to double up its rate. Okay, similarly if it is 27 degrees Celsius, suppose we decrease the temperature of this medium, this temperature 37 degree, it is reaching to 27 degrees Celsius. So in this temperature the rate of the reactions become half, and whenever the temperature from 27 degree it is reaching to 37 degrees Celsius, so in this temperature the rate of the reactions get doubled up. Okay, so this point also you have to remember. After that, let us move to the next, that is pH. So what is happening in the pH? In this pH, same type of graph you can see. Okay, suppose so the second point is the pH. So in this pH point, if you need to see same type of graph we can see. Suppose it is one enzyme. So there is one enzyme I can give you example of. Suppose this enzyme is pepsin. Okay, so this pepsin works in a temperature that is 1.8. Below this temperature enzymes do not work, above this temperature enzyme do not work. Okay, so it is for pepsin. If I give you another example, that is suppose salivary amylase. So for the salivary amylase, this enzyme will have this pH at what pH this salivary amylase works. It is 6.8 pH. Okay, and suppose there is another enzyme, this is trypsin. Okay, right, suppose if it is a trypsin, then for trypsin it is 7.2. Okay, so this is pepsin, this one is salivary amylase, and this one is trypsin. Okay, so trypsin is a very important enzyme in the small intestine. So this pepsin that helps in the digestion of protein, salivary amylase which help in digestion of starch in the mouth, and trypsin that helps in the digestion of different type of food components in the intestine. So what is happening for different type of enzymes, there is a specific pH we can see. Okay, so this is for the pH. So optimum pH. Okay, so optimum pH for different enzymes are different. But what we have seen for the temperature, for almost all kinds of enzymes works in a specific temperature. So optimum temperature for all kinds of enzymes. In case of human body, suppose you are taking the example, then the optimum temperature for almost all kinds of enzymes are same. But within human body, all kinds of enzymes work in different pH. So the optimum pH for different enzymes are different, but the optimum temperature for almost all kinds of enzymes are same. Okay, so these are the two statements you have to remember. Sometime from here also questions can come like assertion reason type of questions. So you have to understand deeply what is the meaning of it. Okay, so you can get these graphs. Similar type of graphs you can see here. We can see enzyme activity. Okay, and here we have here also it is enzyme activity. Here we have put the temperature, and here we have put the pH. Okay, sometime you can be given only the graph where in x-axis and y-axis it will be changed to, in y-axis enzyme activity and this temperature or that factor will be given in the y-axis. Okay, sorry. Yes, the two graphs can be given and you will be asked that which of the graphs is correct. Okay, suppose it will be changed to like this. Okay, suppose here it is temperature, here it is enzyme activity. So from A and B, which is correct? This one is correct. In x-axis we need to have this factor, temperature or pH or substrate concentration, and in the y-axis we need to have the enzyme activity. So these are the two factors that are affecting on the enzyme activity. The third one, which is going to be very much important, is the substrate concentration. So let's just move to the substrate concentration. So after that, the third factor we need to know, this is going to be very much important, and from here the question generally comes. We need to know it very clearly. We have to be very much clear about this concept. So the third factor which is going to act or affect the enzyme activity is the substrate concentration. Okay, so what is the substrate concentration here? See. Okay, so first of all, what is the substrate concentration now? Okay, so what is it? First of all, what are the substrates? Here, this is the enzyme with which the substrates are going to attach. Okay, so this is the enzyme, this is the substrate. Then after that we will have the enzyme substrate complex, then enzyme product complex, and then the product will be ultimately removed from the enzyme. So this will be the substrate with which the enzyme is getting attached. So we also need to know another point, that at what medium the enzyme get attached to the substrate, right? So enzymes will attach to the substrate only in the aqueous medium, in a medium that is having water in it, and in that process, how the enzyme get attached to the substrate? By the process of diffusion. This thing you have to know, that enzyme attach to substrate in aqueous solution. Okay, and what is the process by which the enzyme attached to the substrate, or enzyme binds to the substrate by the process of diffusion? This point you need to know, and always you need to remember the substrate concentration is always more than the enzyme. Enzymes are very less. Okay, suppose you are saying about enzymes are 6 or 10 or suppose 100, the substrate will be thousands or lakhs. Okay, so these are the 3 points you need to know. Okay, so what will happen whenever enzyme gets its, or suppose substrates, if you're having less substrate, it gets to bind with the enzyme, and what happens? See, suppose we have the constant number of enzyme. Okay, so this is suppose 3 enzymes. Okay, first of all, what we have done, these are the enzymes, these are all enzymes. Okay, this medium is a aqueous solution.